Meiosis is used for just one purpose in the human body, which is the production of gametes, sex cells, or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. So basically, in summary, meiosis in humans is a division process that takes us from a diploid cell, which is one with two sets of chromosomes, to haploid cells, which is ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and an egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set, which is a new genome. During the first phase, prophase 1, differences from mitosis begin to show. As in mitosis, the chromosomes begin to condense, but in meiosis 1, they also pair up. Each chromosome carefully aligns itself with its homologous partner so that the two match up at corresponding positions along their full length. Then, crossing over occurs. Crossing over is an exchange of genes. The genes are mixed up, which does not result in a perfect duplicate like mitosis. Also, the nuclear membrane dissolves and centrioles move to the poles, while spindle fiber is starting to form. The DNA has been replicated and then forms the chromosomes made of two identical chromatids, and then the homologous chromosomes line up to form 4 chromatids, or a tetrad. After crossing over, the spindle begins to capture chromosomes and move them towards the center of the cell, the equator. Each chromosome attaches to spindle fiber from just one pole of the spindle, and the two homologous of a pair bind themselves to spindle fiber from the opposite pole. So, during metaphase 1, homologous pairs, not individual chromosomes, line up at the equator for separation. Anaphase 1 is where segregation occurs. In anaphase 1, the homologous chromosomes are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and do not come apart. Finally, in telophase 1, the chromosomes arrive at opposite poles of the cell. Once it is complete, each pole has a haploid number of chromosomes. Then, cytokinesis, which is the division of the cytoplasm, occurs at the same time. At the end of telophase 1 and cytokinesis, 2 haploid daughter cells are produced, each with 1/2 the number of chromosomes of the original parent cell. At the end of telophase 1 of meiosis, the cell enters into prophase 2. In prophase 2 of meiosis, the following events occur. Chromosomes condense, the centrosomes move apart, the spindle forms between them, the nuclear membrane and the nuclei also break up while the spindle fiber begins to capture chromosomes. These chromosomes do not replicate any further in this phase of meiosis. Then they begin to move to the metaphase 2 plate, which is at the cell's equator. In metaphase 2, the chromosomes, or 2 chromatids, line up individually along the metaphase plate at the equator. The 2 sister chromatids of each chromosome are then captured by spindle fibers from opposite poles. In anaphase 2, the sister chromatids separate and begin moving to opposite ends, poles, of the cell. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. In the final phase of meiosis, telophase 2, nuclei form at the opposite poles and the nuclear membrane is starting to form. Cytokinesis, which again is the division of the cytoplasm and the formation of 2 distinct cells, occurs, which splits the chromosome sets into new cells, and then forms the final product of meiosis. 4 haploid cells in which each chromosome has just 1 chromatid. In humans, the product of meiosis are sperm and egg cells. And those were the phases of meiosis 1 and meiosis 2. Thanks for watching.